Hello guys, our topic for today is microarray, the uses and application in genomic analysis. Here is the overview of what we're going to discuss in this video today. What is microarray? Microarray is used for genome-wide expression studies, and it consists of tiny amounts of a large number of single-stranded DNA fragments representing different genes fixed to a glass slide in a tightly spaced array. It involves the hybridization of cDNA molecule to the cDNA template, and the expression level of genes is indicated by the amount of cDNA hybridized to the DNA template. Next, we're going to move on to the basic technique of using microarray. First, we need to isolate mRNA from tissue of interest. Next, cDNA is generated from the mRNA by reverse transcriptase, and the cDNA is then labeled using fluorescent dyes. The cDNA mixture is then applied to microarray, and this cDNA will hybridize with any complementary DNA. Next, the microarray will be scanned for fluorescence, and each fluorescent spot represents a gene expressed. Move on to the application of microarray. Microarray can be applied in genomics and proteomics, in medical biology, in medicine, and in plants. The example of application in genomics and proteomics are SNPs, forensic detection, gene expression profiling, and cell separation, whereas the example in medical microbiologies are microbiome detection and identification, comparative genomics, and microbial typing. The application of microarray in medicines are gene expression profiling, diagnosis of genetic disorder, diagnosis of infectious diseases, and detection of pathogenic microbial agents, while the application of microarray in plants is crop improvement. For the example of microarray analysis, gene expression profiling for cancer research and pharmacological are chosen. But before that, what is gene expression profiling? It is used to monitor global gene expression profiling of cells under a condition of interest. It can be done by measuring the relative amount of mRNA expressed. In cancer research, DNA microarray can be used to analyze the differences in gene expression levels between cancer cell and healthy cell. Cancer cell and healthy cell are compared by looking at the gene expression in which genes are turned on and off. This is the steps of DNA microarray in cancer research. First, collect the tissue from both healthy and cancer cells. Then, isolate the RNA from the collected tissue. The mRNA from the tissue sample are then collected. The DNA copy will be labeled by using different dyes. The green dye will be applied to normal cell, meanwhile the red dye will be applied to the cancer cell. After that, apply DNA on microarray and scan it. Lastly, the data will be analyzed. From the figure shown, yellow spot represents genes that are expressed in both normal cells and cancer cells. Red spot represent genes that are turned up in cancer cells. Lastly, green spot represents genes that are turned down in cancer cells. From this analysis, it can help to accurately classify tumor, and this information obtained help in predicting the patient's clinical outcomes. In pharmacological, gene expression profiling allowed us to discover a new drug, assess drug toxicity, and monitor its safety and effectiveness. This can be done by observing the changes level in the gene expression in response to drug treatments. It is an important tool for diagnostic tests. That's all for today. I hope you learned something from this video. Have a nice day.
Created using Powtoon.